So for our project on mechanisms of antibiotic resistance, we were assigned transformation, which is a genetic transfer between two bacteria. A bacteria dies and it contains the plasmid, which is made up of all of the bacteria's DNA. When this bacteria dies, the DNA is released and broken up into what's called the naked DNA. And this naked DNA contains the genes that make up the bacteria and can contain genes which are antibiotic resistant. Now, other bacteria can salvage this DNA and incorporate it into their own using that new genetic information and become antibiotic resistant.
So this bag represents a bacteria with antibiotic resistance, and the oil on top of the bag represents the plasmid of the bacteria with antibiotic resistance. So we are going to kill this bacteria, quote unquote, with this knife, and this is an important part of the process because the bacteria has to die in order for this process to occur. So as we poked a hole, the contents will leak out into the system along with the plasmid.
So the oil has bubbled right now, and that represents the naked DNA in the system. Now this bacteria has died, so we have another bacteria, which is represented by the sponge, which is going to come into the new system and absorb what's in the system now and incorporate that into its own DNA. So the sponge has absorbed both the water and the oil, which is representing the naked DNA and the other genes from the other bacteria, and it's incorporating it into its own DNA.
Chromosomes, for that matter.